So we made this brain visualization that's based on some ECOG. What's that? How do you guys feel?
Yeah. Yeah, are you?
Sure.
Don't feel pressure.
You okay? Yeah.
No, it's okay.
Okay, so we created an interactive visualization for looking at 3D brains with electrodes on them and time series data. And so one nice thing is it's actually running on Binder, so you can try this out on your own computers if you just go to this GitHub, Matan's GitHub, and then click on Launch Binder, which is down there. And I'm actually going to open the one that I have launched locally. Okay. So, oh, this is going to be a little bit awkward. So do you want to talk about like the widget stuff?
The implementation wise?
Yeah.
Right, so we use neuropathy to present the brain, and then the electrodes are presented with IPI volume, and we use IPI widgets to control different things. The data is basically a patient either making sounds, so speaking, or listening to sounds. So listening.
So let's try listening.
And
then
so this is more of an auditory area, so it responds more to listening than to speaking. But if we look at the more motor area, like the motor strip,
hopefully that works. I didn't click the right thing. Let's see, it's a pretty wide area.
so we get some activation for speaking, but less so for listening.
Yeah. Yeah, and that's it. And so you can, all of the plots update based on what you choose from the drop-down menu. And I think what we're trying to figure out now, so if you had your data as free surfaces, then this can load into this tool. The data itself is just in a dictionary indexed by condition, and they're just NumPy arrays where it's number of electrodes or channels by time. And so right now it's just designed for this one data set, but I think for people who are interested in contributing, we could figure out better ways of loading in data. For example, if you had an event file from something like MNE and you want to take epochs of data and put that in here more automatically, I think that would be very cool. So,
if you have conditions, it will just come automatically to that Dropbox and drop-down menu. And you can also see areas in the drop-down menu at the top, so you can select also like that instead of with lasso and mouse. We tried to, if we had time, we would make the electrodes instead of blue to be the parrots, but we didn't manage. So.
yeah.
So IPI volume has this very useful feature of presenting dots as cats. So we're considering using this option.
Yeah. And I think we should thank also Noah, who is not here, but he was very helpful, and he updated a few things in his package so that we could implement this well and visualize the depth electrodes.
Yeah. So if any of you have been using neuropathy, he did update it just this week to include transparency of the mesh. Yeah. So if you want that feature, make sure to upgrade your installation.
So there is neuropathy 10 now.
Yeah. And that's it.
Cool.
Thanks.
Next is NeuroHackademy. Yeah, I can plug your laptop. Do you mind if you're live with it?
No.
And before you start, come stand over here.
Coming.
Jake.
Do we all fit?
So. All right, so. Okay. I can make it a bit bigger.
Is it big enough? Or do you want to just go with the map full size?
Yeah, but you can start giving the general introduction.
All right, so we're NeuroHackademy Group, and this afternoon we'd like to take you through a journey through space and time, your own space and time. So we were really interested in mapping geographically where we'd all come from and where we'd all gone across our progression. And so we've got a combination of different visualizations which different members of the team have worked on. The tools that we were using, pretty much everything is in Python, which is really, really great because none of us had used it before.
Yeah.
So thank you so much for all the support and help that we've had along the way. So we predominantly used GeoPy, Leaflet and Folium to create the visualizations, and then there's some stuff in Giphy as well. Okay, all right, so Jeff.
So yeah, the first thing that we wanted to try to capture and to visualize was basically on the basis of your responses to the questionnaire where everybody was born, so the hometown. And so we wanted to be able to plot on this map, in terms of dots where everybody's hometown is, and you can see the distribution, and you can also, for example, go on one data point and ask where this was, and it's going to tell you the actual location. So this, we did this, then we tried to capture how people moved throughout their career. So we went from hometown, we asked where you did, for example, undergrad, and we can plot different set of points. Then where you did, for example, an RA or lab manager position, where you did the master's, PhD, postdoc, and for the few data points of faculty, there is also some of that. And now when you plot everything together, it looks a bit messy, but if I clean it up, one or a couple interesting things that you can see is that, for example, from going from hometown to, you can see how people are really spread like across the world. But then if you look at where people did the PhD, their PhDs seems to be mostly localized, like in North Europe and the States, and that's even more true for postdocs. So if you just compare hometowns to postdocs, you see how much more localized that is. Faculty as well. So anyway, this was just something to capture on different levels where people moved across their career. And another thing, and so this was very much a collaborative effort. I wasn't the only one obviously working on this. I received a lot of help from my teammates, and obviously Tal and Chris and Ariel as well, and Kirsty. So the next thing that we tried to visualize, Antonio was working on that, and it's basically, you can tell.
Yeah, so
I'll just scroll down.
Thank you. So in the previous plot, we had kind of a lot of dots, so you can see kind of where people are, but you could not necessarily see who moved where. So we tried to do a bit more of kind of showing you who moved, well, not necessarily who, but like kind of what distances people moved to and from. So this is just showing where people were for their undergraduates, where they were born, and then where they moved for their undergrad studies. The one thing that we could not make work was an animation, so you can't necessarily see in what direction they're going. You can just see like a line is, so for example, you can see the hand move from like the UK to Australia, but you wouldn't know the direction if you didn't know who it was. which was a little bit unfortunate, but we still think it added, an extra level of clarity. And then, if you can just go. So we added that also this is kind of where people went to undergrad, and then those who did like a lab manager position or an RA position, this is kind of how they moved. And then we also plotted it for the PhD. So here we took either where people moved from RA position to PhD, or if they did not do an RA position, like from their previous point, so either undergrad or the place where they were born. So that was kind of a more in-depth visualization of the movement and the distance.
Okay, so also related to distances, then we tried to quantify. You can tell more about it.
Yeah, so basically how many miles did people travel between different stages and also overall throughout the whole career. And yeah, basically I calculated this thing separately, and here you can see for different career stages. So like the first violin is home to undergrad, so it's mostly like not so far apart from that one outlier who is at 12,000 miles, and this is Hannah moving from UK to Australia, so that's 12,000 miles. But then you can see that maybe PhD to postdoc, the maroon one, it's probably the longest distances overall. So that was the biggest move. And yeah, postdoc to faculty is kind of less than 2,000 miles.
Next plot.
And then I just calculated how many miles people traveled from being born to their final destination. And yeah, on average it's around 3,000 miles. And then there was one person who moved 0 miles, so who just stayed for their career in their hometown.
Okay.
Yeah, and then again 12,000 miles, it's kind of on top. But most of us cluster below 2,000 miles.
Okay.
And that's it.
Thanks. So then Damien was working on a number of different visualizations. Which one would you like to have?
Let's go ahead and do not that one, the third one maybe. Yeah, that one.
Okay.
Yeah. Cool.
Oops.
Oh well. So that didn't work out so well. But I tried to see with a spring graph how certain things worked out. So the first thing I did was faculty versus non-faculty. And when I just randomly did an algorithm to spread it out, the faculty made this crazy shield. And you already saw, I was wondering what we were being shielded from. I don't know. I was going to try to make a bad science thing, but it was too long.
You can make full screen.
Oh yeah, here, how do you do that? Cool. So in any case, you can see the data points, and every one of us that's not faculty, we're connected. This was just binary connections. But then I wanted to see if there was something more informative. So yeah, let's do this one first. So I did a circle graph where I first did modularity, and then I did how many interests we shared. So what this is it's your interests that you listed. It's kind of sloppy because people use different terms. So if you did neuroimaging and fMRI, it wouldn't match. But I took all the lists of terms you said you were interested in your recent focus, so faculty or PhD or postdoc, whatever. And these are, the weights are how many you share in common with everybody else. So then I did modularity, threw us into some groups, and then the lines are just those weights based on how many interests you share. And it's funny because I didn't get the words up there to look because you have to kind of dig into it, but see, a lot of these are grouped like fMRI or whatever. But there's this one crazy outlier up here, which I want to call out, which is Chris, because he thought it was hilarious. It's just because he didn't list anything in the same phrase, right? He had overlapping interests, but it was this just quick and fast way of doing it with actual word matches. But then, But then I think this is funny too. Then I visualize it as just a normal spring graph. And again, we have the spring graph with all of us here, and Chris not connected to anybody. If anyone knows much about spring graphs, I had to change the gravity. I had to make it really, really high to pull him in. So in any case, I thought it was an interesting way of seeing how we grouped using sort of a modularity clustering view.
All right. And the final visualization type was performed by Johnny, and wait. It's here. So yeah, maybe you want to tell people.
Yeah, promise. This is the last one. So we are just trying to capitalize people's research interest in a different way using word cloud. And maybe some of you already know what that is. So basically, the larger the word font is, the font size is, represent the more frequent the word is mentioned. across all people. I just plot the PhD research here as an example. So we use a head and then use a brain. If you go further down, I also do separate by continent, but it's quite similar. We've only got so most people work on fMRI, neuroscience. Yeah, we've got a few samples from Asia Pacific, and then maybe go for last one, faculty.
Oh yeah. So just go all the way down.
Yeah. So I also try to split the research interest by people. By their academic stages.
Right. So this would be.
So this one would be interactive. So from undergraduate, research assistant, master's, and PhD.
And yeah. All right. Yeah.
Yeah,
I think
I think that's all. Yeah.
Next we have the Degenerates. Degenerate Group. Oh, do you want me to bring it up from here? Okay, yeah, I can thank you.
Thank you.
Oh, yeah, okay. Is that here? Okay. I think so.
So, hi everyone. We are the Degenerate Group, and we're going to tell you something about our awesome hack project that we did this week. So, first things first. Does this work? Not really.
Sorry. Try to see if we can coax it into working.
Yes, thank you very much. First things first. Who has seen or used or heard about this app? Amazing. So everyone is on the same page. This app was really popular up to, I don't know, a couple of weeks ago or something like that. And it's pretty cool. It's amazing. I mean, they use models to kind of force pictures to use another shape. And in practice, in this situation, they oldify the faces. So we thought, well, why not try that with the brains? Let's see if we can get brains to get older and to be similar to an older brain. To do that, we had three main stages. And first of all, we needed to collect data. We used some open source databases. And then we had our modeling part. So we first tried to model with the CNN whether we could classify older and younger brains. And in parallel, we work on a generative adversarial network and to try to produce from younger brains images of older brains. And then we had our evaluation stage, which is not totally completed yet, but I mean, we got started. And also that stage was divided in two. So we first wanted to try and see whether our previously trained network were able to classify the new images correctly as older brains. And secondly, we wanted to extract the volume, measure the volume of the gray matter, and see whether the distribution of the fake images gray volume was more similar to the older distribution or the younger distribution. And so, about data processing.
So these methods require a lot of data, and we used two data sets. We used a young data set, it's data from the HCP project, and we selected, or we took over 1,000 T1 weighted images. The age range is between 22 and 36, and these data sets were on the Amazon S3 bucket. And for the old data set, we use data from the Oasis project, also over more than 1,000 T1 weighted images. Some are cognitively normal, and some show some cognitive decline. Age range is between 42 and 95. Thanks to Steph Rossi for granting us access to that data set. So the input data to work with is, so we made some Python scripts for converting nifty files to PNG images. That's it. Okay. Now the how part.
This is actually mine. Right, so sort of half of this project was, again, just kind of a silly thing in scope, in that we just wanted to try to classify the age distributions of HCP patients using simple convolutional neural networks. So the HCP data has some privileged access to their exact ages, but the non-restricted demographic information bins these patients into age ranges of, 22 to 25, 26 to 30, 31 to 35, and 36 plus. So we just wanted to see if we could classify those simple age groups. And we had just one midline slice in the axial plane per subject. And we also did a post hoc analysis, which was a related question of whether or not we can classify processing speed of these patients using the same data, just using a median split of their scores. So we built a very simple CNN architecture that looked at various patches of this axial slice and went through a series of simple convolutional and max pooling layers that resulted in classification of these subjects into one of these various age buckets. But unfortunately, as you might guess, this really didn't do well. This plot here on the right is just showing the training versus the validation accuracy, which hovered at exactly chance, about 25%. But overall, we all had a lot of exposure to using AWS, Keras, Python, Git, and Docker, which is kind of what we got into it for. Who's next?
I was speaking over the CNN architecture thing, so yeah. On top of this CNN architecture, I tried to use transfer learning. I think you're kind of familiar with it. I used ResNet architecture, which is one of the very old revolutionizing techniques of making convolutional neural networks a little bit robust and fast. And using the ResNet, I fed the input from HCP data and the Oasis data, and tried to generalize simplify the classification thing. And our aim is to classify the young and old, and using different datasets, and it worked pretty well on training and validation and test accuracy of the same kind of images. But we also tried to test on the gun-generated images, and it drastically and finally failed. You can see my results on I shared on the Slack. I couldn't put the slides because of the hurrying thing. And should I move on again, or switch over?
Yeah, I think it's my part. Yeah.
So yeah, actually, Meltem and I, we wanted to organize a breakout session about GANs that we didn't manage to. So just to give you like a meta understanding of what GANs are actually doing. So GANs basically are a combination of two networks, in our case two CNNs actually, which are kind of playing a game against each other. So one of the networks is a generator network and the other one a discriminator network. The generator network gets some source data and has the task to produce some synthetic data with specific characteristics that you decide. And this synthetic data, or part of the real data, gets then fed into the discriminator network, which has the only task to decide, okay, the stuff that I'm seeing right now, is it real or fake data? And then when it does it, and of course you can use this, for example, to translate from image to image, for example, from if you have real data that looks like this, like flare data, and some data that looks like T1 data, you can let the generator produce something that looks like a flare, for example. And in the first iteration, the generator might do like a very shitty job, like a lot of noise and stuff like this. So the discriminator easily finds out, this is fake data, I know that. And then feeds this information back to the generator, who then also improves its performance and thereby improves the output of the synthetic data. And like this, these networks are like playing this adversarial game against each other until they reach some kind of Nash equilibrium in this game. So the discriminator cannot clearly distinguish anymore between real and fake. Yeah, and so this you can use if you have like paired data. So if you have like, if you would have for every subject a young image and an old image, we could have used this architecture. The problem is there's no data set available which scanned people 30 years ago and now, obviously. So we had to use another architecture which is called CycleGAN. I think, Meltem, you wanted to explain?
Yes.
Yeah.
One thing I really want to point out is from here, actually generator is born blind. It's fed with only random noise, and it tries to get some feedback from discriminator, which is able to see both kind of distributions of data, and it hallucinates data. Basically, it's like that. And in GAN, there's a cycle consistency loss tracks such hallucination. And in this architecture, there are two GAN architectures, the two generators, one of which is generates, imagine X is young brains, Y is old brains, and one generator generates hallucinated fake young brains from the input of the young brains, which is not the generally GAN's input. And another GAN also generator tries to do same with old brains, and they are with the cycle consistency loss, they are exchanging information between what they generated. So they are not interacting with their data input, they are just exchanging the dreams of the GAN. And eventually it produces results, and the important thing is this is unpaired, so which means we didn't track young people and weight them and make them old. We have some young people unrelated to old people. So this is unpaired. So you can do it if you have pictures of flowers. You can't do doodles of flowers. You can just feed the cycle again. You have doodles. This is unpaired. So this is the difference of the idea, and I think it's all.
Okay, it's me again. So now we come to the actual image results of our GAN. So on the top row you see the real young HCP data, and on the bottom row you see the fake old data. And what you see is that there's definitely some noise happening, but wait, can I see? Yeah, okay. I don't see the mouse here. So, but in some parts you can actually see that it does, to some degree, what we wanted to do. Like, for example, if you look at these two gyri in this subject, you see that it's like trying to increase the amount of CSF in this part. In general, they look a little bit more unhealthy, to be honest. They also just look a little bit more ugly. But yeah, you see here in this like a cleft between the hemispheres that it tries to increase the overall CSF. What it doesn't do so much is like increasing the ventricle size, which we were kind of hoping for. Also, it seems to have some problems with bias field. So you see here's like a giant bias field from bottom to top, and it just here deletes the whole cerebellum. So apparently that's what's happening when you get old. Then yeah, these are some more examples. I think this one is kind of impressive because you see here that it's like kind of trying to do some atrophy in the cortex. Yeah, we have quite a lot of images, so in total I don't know, like 200,000, 300,000 images or something like this. We also made them to Nifties and we tried to do some analysis on them, but everything took such a long time because we had so much data that every copying step is a time critical. And also, I want to explain this noise a little bit because these here are actually fake young images. So this is basically what Melton tried to explain to you is that this cyclic GAN basically has two generators. So it tries to produce also out of the fake old data back some fake young data, correct? Yes. And you see the problem here is that on the one hand, it just doesn't work very well. And on the other hand, if you look at the image to the right-hand side, this one, you see that this is actually an axial slice, although we fed it only coronal slices, or we wanted to feed it only coronal slices, which doesn't mean that it produced this by itself, which would be impressive. What it means is that the OASIS data set actually is not very well aligned, so you have a lot of like misaligned images, and yeah, it's just very, very messy data, very messy data set. So yeah, I think this would be something to improve. All right.
I guess the final step that we didn't quite get to because we spent so much time copying files and morphing them into PNGs was to actually do an analysis on the volume. So typically older brains have like less gray matter essentially than younger brains. So what we thought would happen with our amazing results is that gray matter volume would be super high in younger brains. And then it would be less in the OASIS data set, and we would have less gray matter also in our fake brains. So the results are being processed at the moment, so I guess watch this space. And if you want to get involved, just join the DLGAN channel on Slack. Oh yeah, and this graph does not exist, obviously. And yeah, so yeah, basically this is kind of the end of the presentation. So we just wanted to say we had a lot of fun, we learned a lot, had really good food, and thanks everyone for the help from various people. Yeah.
Next we have the volume.
Someone has a question?
Question?
That's amazing. I don't think you've done that in a week. Could you speculate why your CNN didn't train? Why you didn't got chance?
I couldn't implement the stuff. I blame myself.
But do you think there's come on? Do you think like there's too much noise in the image, or?
Yeah, noise matters, but actually I couldn't. I tried to use all of the tricks, and while I writing it, I missed the timeline, so it's all me.
I mean, size of the data is a big factor. So initially, I think our training data was too little, and then by the time we were like, we incorporated all the data, there were other confounds. So given more time, given more data, probably could have worked.
Potentially, if we bias corrected the data as well, that would be helpful.
Yeah. Thank you.
Volume to HCP 50.
Would you mind using slide to this?
Where is the HDMI cable? Yeah.
Yeah, sure.
That's it.
Okay.
Over there. Come over here.
Yeah.
All right. Hi, people. Yes. Cool. I put Chris's name on just because I don't want to feel so lonely, just two of us. Okay. So we were working on a pipeline and a related feature to put your own volume-based preprocessed data into HTTP SIFT format. So why do we need this? And why is it not moving forward?
Try this. Oh no.
No? Just give me a sec. It's probably just mirroring display. Just mirror display. Monitor on off. Simulation on order. All right. Cool. Yeah, that should work.
Yes.
Okay, so give you some background. So we want to use HTTP surface for our own data, and we want to use it on a even lower resolution. I believe this week I've seen some people struggling with surface-based analyses because the number of vertices is too large. Oopsie, what happened? What happened?
It's just loose.
Yeah, I don't know. It just keeps switching back. I forgot to apply, sorry. System-wide, keep this configuration. Cool. Yeah. And so this leads us into a really wild adventure of digging through horrible code base of HTTP and the lack of Python-based tool for working with HTTP Workbench and GIFT SIFT format. So there are three challenges we face. The first one is using a customized HTTP pipeline to do the final part of mapping volumetric data to HTTP surface. And this is done by me and Jia. And the second one is we just want to plot this surface data in a multi-view montage to make it pretty. And the third one is we want to get a start with the lack of high-level API issue with SIFT and GIFT format. And it's mainly between my discussion and Chris. So let's hand it over.
So I started, in the spirit of hacking, how I was motivated to deal with this problem is it's just a personal pain that I've been dealing with for months. So I've been wanting to project the volumetric data to the HTTP surface because of the superior mapping between volume and surface. And you might wonder why we want to do that. So there are a few reasons why I want the data on the surface. One is it's much easier to visualize. I'm not trained in neuroscience, so it's very hard for me to look at any volumetric data in the volume and think of anything. So it's much easier to interpret stuff on the surface. Second is the mesh of the surface offer a lot of advantage in computation. And that's what I've been dealing with. So one is there are a lot of voxels in the volume that I'm not interested in. So by doing analysis on the surface alone is a much cheaper approach. Second is I can make use of the mesh geometry, such as adjacency between vertices, and it's also a much easier way to perform upsampling or downsampling with the data.
Press space.
Yep. Oh. Oops, sorry. So this is very geeky slide. So basically the whole point is I want to wrap all these HTTP Workbench scripts into Python functions so that I can do everything within Python instead of having to switch between environments. So from some of the tips learned from Thors and advice from Chris on MyPipe, so wrap everything in, with the object-oriented programming style so that it's much easier to handle and to manage the different files that are spread over the place by the HTTP pipeline. And the future plan is not to reinvent the wheel, but eventually to incorporate these wrapper functions into NiPipe. There are some working code on our repo that you guys can check out, but more polished code should be pushed out soon in the future.
All right, and then the next thing we work on is visualization on montage. So the first thing we learn at this 2 weeks is don't reinvent wheels. So the first thing I did to search for existing issues on iLearn, and we found one, but there are some problems. So they currently have a branch that hasn't been merged to a master yet. It is plotting volume data onto the surface. So I was really curious. You're presenting data on the surface anyway. Why don't you just write to accept like surface input as well? So I asked this on GitHub, and they replied, and please don't take a picture of the next slide because I think it's a little bit mean. The response is, the goal of the function was to accept volumes as input. Surface was never planned. Surface was never planned. I was like, this is madness. I cannot accept this to happen. So we wrote a script to achieve what we want for now. And because this script is not optimized yet, so it takes a bit time to run, but the result will look like this. So this is the data I have for one participant running through the modified HTTP pipeline and plotted on a very low res surface. Yeah. So it's kind of working, and the future plan is we're going to create this function, a function for this plotting script, and open a new issue on iLearn to add this new feature. And hopefully, this will lead to profit. Yeah. So after you put all the data onto HTTP surface and you do some fancy analyses in Python, and you'll reach another issue. I can only view it in like the fancy function I wrote for surface montage. What if my boss doesn't use Python and he has a problem in viewing and he wants to use Workbench? So this question led us to the current chaotic state of GIFT API in iBabel. So I'll give you a few examples about how we work with it. So right now, if you just load a GIFT file in, you need to access D arrays in order to see what is inside. And who can tell me what are those things? I have no idea. So I have to do the next thing is I go into each of the array and then print summary to know, okay, so this is an intent called point set and it has some stuff in there, and I'm still not sure what it is. Yeah. And if you want to access the data that is related to that metadata, you need to use data to access it. Oopsie. How do I go back? Nope. Oops. So, yeah, and then you will see, oh, okay, I probably know what this is now. Maybe. Yeah. So it gets even more annoying if it's a functional GIFT file. And I'm not going to print out the content of this one because, as you can tell, however many arrays you need to put into the GIFT file, however long that list is going to be. So if I have a resting state data of 9 minutes with a 3TR, it will just give you 180 things in that list. And in order to get all the data out to do some analyses, you need to go into each data array to get them out. And so now you know how difficult it is for loading the file. Now we are going to talk about how even more annoying with saving file. So the first example is kindly provided by Sam, and this is a function he used in his research. What he did was to get an existing GIFT file and then just strip off the data, maintain the metadata intent in it, and then add the new data you want into GIFT and then save it out. If you want to do it just in iBabel, you need to know what are the metadata are. Otherwise, the Workbench will not read it correctly. And this is quite fiddly, and it took me about a month to figure this out. Yeah. So the new plan is to propose a new API. Currently, we open the issue on Nightbabble, and so we have two parts. The first part is how to access the metadata, and the other part is how to access the data. And Chris has already replied to it about an hour ago, but I haven't got a chance to look at it yet. Yeah. So the future plan of this is we're going to discuss the API issue with the community, and we are hopefully going to use the experience of working with Jifty to improve the Sifty input and output interface as well. And finally, this will all lead to profit. Thank you.
But we are all of us are developmental cognitive neuroscientists along the lifespan. And so we were interested in trying to use the HCP lifespan data and using NeuroPython for visualization. The purpose, I think, overall of this group was to sort of try to actually work with HCP lifespan data and then like set things up for the future. So we will get started on what our journey was through this week.
Yeah. Buckle up because there's going to be a lot of this.
All right. So we started the week off like really excited. We were like, great. We're going to download the HCP lifespan data set, and we're going to run these pre-processing pipelines, and then we're going to use NeuroPython to visualize everything and look at differences in cortical thickness, gyrification, T1, T2 maps, and that's how we started. And then,
go ahead. So first we got online and we decided we're going to download the HCP developmental and adult data sets, except that we couldn't. So then we thought for a brief second we might use the ABC data, but then we decided, nope, that's not going to work. But luckily we found a small data set. The HCP lifespan pilot data set was released a few years ago. It has 27 subjects in 5 age bins.
Yeah, so they range from age 8 to age like 75.
So that was Monday. That's what we spent Monday doing. And so then we decided we were going to upload the data to AWS and run the HCP pipeline. So I personally have had a little bit of experience with AWS, just enough to be a little scared. And so that was basically what we did on Tuesday.
Yes, and we also spent a lot of time creating a new Docker image, only to realize that we really did not need to do that because HCP already has those things. So just lessons in not reinventing the wheel. But we did learn how to create such an image, so that was exciting.
Yeah. So Wednesday was riding the cloud. We realized whenever we found this Docker image the pipeline that we decided to use, everything had to be in bids. So then we had to bidsify our data. And then we got it running on the HPC pipeline, HCP, that's a typo. We set up a lot of instances on Amazon Web Services to process our data. And yeah, I think we spent a decent chunk of change doing that.
So thank you for that opportunity. Okay, so Thursday was exciting because we actually finally finished processing a single subject, an 8-year-old, and we were able to load it in NeuroPython and visualize cortical thickness. So this is a heat map, although it's a cool map because it's looks like a galaxy, and that's kind of fun. So that was really exciting, and we got to load that, and so
maybe.
Yeah.
Oh, there we go.
So Kendra's going to walk you through. You just have to drag it over, I think.
Oh, you can't see it. Sorry, guys.
So she's dragging over in our Jupyter notebook, loading in a single subject that we pre-processed, which the HCP pipelines, that's an 8-year-old, and visualizing. This is showing their cortical thickness.
This is
histograms.
Left and right, nice and symmetric. Keep that in mind, because the other ones aren't.
And then this is showing you a heat map of the cortical thickness for that particular subject. So this is an 8-year-old. Wait, do you want to slow down?
Okay, sorry.
So that was super exciting. You want to continue along the notebook?
Sure. This is the same image, just plotted on an inflated brain and using the cool color map. And this is just weird looking. It looks like a brain on fire, I think.
I think that's the surface area.
Yeah, this is surface area, but for some reason it looks like this.
Yeah.
There's a little hot spot there. Be careful. Okay. Be careful. It's on fire. So this is at the other end of the age spectrum. So one thing you can see here is the gyrification, right? It's pretty apparent even here. This is fun. This particular subject, for some reason, has different atrophy in one hemisphere than the other. We don't know if that's a processing error or if that's actually real.
It's a pretty big difference.
You can see it really clearly here.
It seems like that might not be correct, but you know, it's kind of interesting just for the visualization purposes to see that. So same image this time.
A cool map.
Inflated. And yeah, the brain, it's burning. Anyway, all right. Now I have to figure out how to get out of this. Just go here. Oh, where's my? Sorry, guys.
Where's your mouse?
There it is.
Oh, yeah.
Got it. Yeah.
Okay. So. So then we tried to do some things. Yesterday was a little bit frustrating. We learned sort of the pluses and minuses of cloud computing. We also sort of learned how to allocate memory properly in the way that we're running our functions. As Ariel pointed out, we broke the cloud, but then he fixed it for us. So that was up until like yesterday evening. So we were trying to, for the purposes of sort of proof of concept for today, we wanted to at least run one group analysis by age across the brain. It turned out yesterday that in our loops we were overwriting one hemisphere with the other, so we only have data for one hemisphere, but that's fine. Another lesson. So today we were actually able to finally run our analysis across, so sort of register everybody to FS average and run just a linear regression looking at age across all of the vertices in the brain and look at cortical thickness. And, well, actually, if we have, hold on, there it is, maybe, yeah, so you can just scroll down, and this is showing you the group map. So this is just the T statistics looking at cortical thickness as a function of age across the brain. So this is, again, from kids 8 to adults 75 years of age. Obviously, we would also want to look at nonlinear functions here, but this is sort of just a starting point to be able to look at the HCP lifespan data and structure of the brain. Then, all right, so what we learned and where we're going. So we learned a lot about Amazon AWS, about Docker, about how to use Jupyter notebooks, and about NeuroPython, and a special shout out to Noah, who is an incredibly helpful resource, like really so helpful, even when he wasn't here. And we really look forward to hopefully soon, so the first release of the HCP lifespan data is out, but it's not currently on AWS. And so that will make life a lot easier. So when it gets released on AWS, we really look forward to kind of working with the entire data set and doing more analysis. And so we're also interested in doing analyses beyond these structural analyses, looking at other types of structure like gyrification, T1, T2 maps, and there's resting state data, task data. And we're also interested in looking at other kinds of demographic data. So if you're interested in getting involved, let us know.
You want it back? Do we have time for that?
No, she just wants.
She just wants to take a picture of Hackers Gonna Hack.
Next are the flyer annotations group, or NeuroHackademy. Stand over here. Photo?
Ready.
Yeah, you can talking.
I'm not going to do all of the talking.
Yeah, that's fine.
No, I'll let y'all do some of it. Does anybody know how to make these Jupyter presentations full screen? Make the top go away?
Yes.
You said use full screen?
I see exit full screen. I'm in Firefox, not in Chrome. Cool.
The answer, you don't.
I'm cool with that. All right, so we started out as the NeuroScout group, and then we realized that NeuroScout doesn't do what we want it to, so we switched to being the Flyers group, because that's really what we wanted to do. So what we were really interested in was all of us in some way or another either work with or interested in working with naturalistic stimuli, and we really wanted to figure out how to extract features and really just mess around with Flyers, because it's sort of this kind of was interesting package to interface with some of these APIs. We just all kind of wanted to know how it worked. So our main goals is we wanted to familiarize ourselves a little bit with Flyers and the NeuroScout sort of architecture and infrastructure. And then we wanted to get some comparable features like words, faces, objects, these sorts of things that we can maybe compare quantitatively or at least just sort of look at them and see how well they match with one another. And then we also wanted to visualize some of these between APIs and if we had some ground truth. So we had like a script that we knew was correct for the movie that we analyzed. We wanted to compare between the two. And then we wanted to do a few pull requests with Flyers to clean up some documentation and just make stuff easier. So this was what most of our week looked like, was doing stuff in Flyers. So this is the movie that we chose to do. We picked a sort of iconic scene from Forrest Gump that hopefully you'll be able to hear. Let's see how it's It's because my computer is muted.
Thought I'd try out my sea legs.
But you ain't got no legs, Lieutenant Dane.
Yes, I know that. You wrote me a letter, you idiot. Well, well, Captain Forrest Gump. I had to see this for myself. And I told you if you were ever a shrimp boat captain that I'd be your first mate. Well, here I am. I am a man of my word.
Okay.
Yeah, but don't you be thinking that I'm going to be calling you sir.
No, sir.
So if you don't remember that scene, the boat crashes into the pier because he had jumped off of the boat to say hi to Lieutenant Dan and forgotten that he was the one piloting the boat. Cool. So we will let one of you talk about face extraction since that's started.
Okay, so Adina and I worked on face extraction, and we actually tried 3 different APIs, this face recognition extractor that's built in Pliers, the Google Vision, and Clarify. And our aim was to visualize these faces and then compare them. We got partway there before I made a terrible Git mistake. Yeah. Okay, so here is a vastly downsampled version of the video you just watched with the faces annotated. This only has Google and the built-in. It's quietly downsampled. Blue is the built-in, and red is Google. And it's pretty interesting that Google misses quite a bit, actually.
Thought I'd try out my sea legs.
But you ain't got no legs, dude.
And then we tried to compare this a little more quantitatively.
Maybe.
With a different video.
Well, that was the first thing we tried it on, and we would have presented the real movie, but it was just 5 minutes too few time. So this is
Today, after 2 years of negotiations,
comparison of all of these
the United
tools
States,
with the faces
together with our international
annotated
partners,
and the difference in size, for example, that all of these tools record given the face sizes. Yes. That was it, right? Yeah. Nothing more.
Cool. You want to talk about speech to text?
Yeah. So next, or kind of concurrently, we worked on speech to text. So what you have to do, or actually there's 2 ways to do it, but you can extract the audio. So we converted the audio and then extracted the text using Google Speech's API. And then here in some Vader sentiment analysis on it. And I worked on kind of closed captioning it to check how well it was captioning our text and kind of how accurate it was with timing. And so comparing the kind of script to what we got, it was pretty similar. You can kind of calculate a cosine similarity to see like kind of what the overlap is between the two. And it gets some things, it misses some things. We also noticed it missed a lot of things due to Forrest's accent. So a lot of that was kind of like you would have trouble with it too if like Dan's sounds like Din or something. That's the...
So this is the Vader sentiment analysis, and then we have, let's see, do we have that closed caption video?
Oh, there it is.
Oh, yeah.
So you can see it calls like Lieutenant Dan, Lieutenant Di. But otherwise it's pretty accurate and the timing's pretty good if you listen to it at the same time, which is, I would consider a pretty good success.
Yeah, pretty good.
You can go back to your sentiment.
Yeah. So we were also interested in running some sentiment analysis, which as Tal sort of alluded to is pretty fantastically bad. Vader didn't do a good job like at all. Granted, the scene was not super sentiment heavy, and it's a short scene full of mostly like sort of normal stop word type words, but it only picked up a few words with positive sentiment. So this is just a depiction of well, it's a 1 if it was positive sentiment, and it was 0 if it didn't detect any necessarily positive sentiment, and the x-axis is just each frame of the movie moving forward. And then I also scored, we're currently developing, have a preprint out in the lab of a tool to pull out words related to moral foundations, like for moral foundations dictionaries. And so we ran that on it as well. This is just a time series of the rolling average of care words, so like from the care foundation. And you see as it moves over time, there are some words relating to caring for what people are doing, these sorts of things. So then the last thing that we did was object detection, and this had a really similar workflow to the face detection. We wanted to downsample the movie to 2 hertz just to not blow up Google, and then so that just 2 still frames per minute. And then we first used a sort of built-in extractor with TensorFlow that doesn't have to actually call out to an API. And then we also used the Google Vision API extractor just to sort of see if we could get a comparison between the two. And you'll see that actually the comparison between the two turned out to be fairly difficult because they didn't pull out anything in common. They use different labels, and Google is like really conceptual, but you'll see that here in a second. So this was you won't be able to read any of these labels, but I can just point them out for you. This was the built-in extractor. And you see things like banister, which kind of makes sense given that there's a pier and some railings and these sorts of things. And then bathing caps and lakeside, which I guess is fairly intuitive. One of the funnier ones is it finds here what it thinks is an ice lolly. And I went back and I went to the movie and looked at where each of these little spikes are, and they're when the frame pans over to Lieutenant Dan's face and he has that giant stogie. And so it thinks Lieutenant Dan's eating a popsicle instead of smoking a cigar. And then the one that I thought was the most funny, oh actually you can't see it down here, but there's down at the very bottom, one of the ones is it says website, and it's really confident throughout the whole movie that there's a website contained within this movie. And Maya had the idea that we think it's because of the bounding box around the movie, where it's sort of like a gray bounding box and then the movie, and it's kind of in an iframe, and it thinks that might be some sort of like maybe a website or something like that, which is interesting. So then this is the Google extractor. You'll see it extracts way more things. And it extracts several things that are more conceptual. So we see things like love and let's see, happy and hair and yeah, several other things. But it also gets like bare-chested when Lieutenant Dan's not wearing his shirt. But yeah, and there was actually no overlap in common labels between the two. So the quantitative comparison, you would have to do some fudging around to figure out like what even you could compare quantitatively between the two. Yeah, so we also did a lot of committing and a lot of PRs to Pliers and to our own repository. There's some screenshots of all of our committing and editing and these sorts of things. And then what we learned is we had 2 members of our team do their first PR in this project, which was super fun. We were excited. And then we also learned that merge conflicts are awful. We had almost a full day where we were all sort of trying to figure out a particularly gnarly merge conflict. And then we learned, and this is one thing that distracted Sam and I for part of the project, is trying to dream up what it might look like to have a repository of the actual stimuli for these naturalistic tasks, because a lot of the times they're copyright protected and people don't want you to access them. And then Pliers does make the feature extraction easier. And then we also learned that APIs are really difficult and they want your money really bad. They really hamstring what you can do without paying them or uploading your stuff to their server. And future directions is we're going to be working a little bit more on APIs and Pliers. Right now, like the IBM API is extremely broken because IBM changed a bunch of stuff. And then also working in like the Amazon Vision API and some other things. And doing a little bit more thinking about how to quantitatively compare between different APIs on certain measures. And hopefully collecting some more ground truth data, doing some sort of, like train some RAs or some coders to go through and pick out some things that these cloud APIs also pick out, to see if we can quantitatively compare them. And then doing some work, Liberty and Sam were interested in figuring out phoneme matching, how you get the actual script to match up with the audio so that you can know what words are being said at what time. And then extracting features from longer stimuli. We really just did this 1 minute of stimulus, so we want to do some more stuff. But yeah, that's it. Thanks.
Next up is meta decoding. the back there?
Yes, I need a Thunderbolt thing.
USB-C?
No, I need the other one. This one?
Yeah, these are to VGA. Does somebody have USB-C to, sorry, no, whatever the previous generation is to HDMI? Oh, you have HDMI here.
I have an HDMI.
Yeah, you have HDMI.
Yeah. Totally forgot.
Come close to our photo.
Okay, now how do I... Oh, where is it?
Okay, so we are MetaClass. MetaDecode is another name that we went by. I think class is more funny, obviously. So what is MetaClass? It's obviously just a classy way to do meta-analysis. So we're done, right? So the inspiration for this kind of, why MetaClass? Why these two sorts of concepts together? Well, we learned this week that meta-analysis examines outcomes from several studies to determine trends in the data. That's sort of where my background is. And then we sat through the talk on machine learning where we got other definitions for it, where machine learning automates model building. It also represents systems that can learn from data and locate patterns in data, and finally make decisions with minimal human intervention. So this got me thinking, right? In meta-analysis, we have data that might lend itself to an approach like this, and so I kind of thought, well, if we take meta-analysis and we pair it with machine learning techniques, could we come up with some kind of a meta-classification, right? Meta-class. So neuroimaging meta-analysis, just a quick background in case anybody wasn't in that wonderful talk that Katie and I gave on meta-analysis. In a traditional neuroimaging experiment, subjects go into the scanning environment and they complete a task. This could be like a working memory task. We note where we see activation in the brain, and we note it in 3 dimensions, so x, y, z coordinates. And finally, when we go to publish, we publish those coordinates as a table, right? We say we saw activations associated with memory task, with 3 back versus 1 back versus 0 back, and so on. In neuroimaging meta-analysis, we go through the literature and we identify these tables across many studies that meet the same kind of criteria. We plot these coordinates on the brain, and then we blur them using a 3-dimensional Gaussian distribution where the extent of the distribution is related to the sample size of each study. And so for the purposes of this project, take the task pain. We can go through literature and identify these modeled activation maps from different studies, and we can sort of decompose these brain images into a long NumPy array, so basically 902,000 voxels, and do this for all sets of experiments that we identify and sort of get the stage ready for a machine learning project. Now I will pass off.
Okay.
One more. And then in sort of the meta-analysis of neuroimaging game, we have 2 big players that contributed to these data sets. The first one is BrainMap. Just a quick background on BrainMap. BrainMap is a database of neuroimaging studies where these coordinates are entered into the database manually. So we have people that are hand curating these studies, reading them, saying this was a working memory study, the sample size was the following, and other features about the study to describe it. And we also have NeuroSynth. NeuroSynth is an automated database, or a database that uses automated extraction to remove the coordinates, features, and stuff like that.
Okay. So the task before us is to decode these activation masks into different task categories that they were assigned, that the label that they were assigned in these databases. So for the purposes of this mini project, we took data from 4 tasks, or tasks that have these words, like Sacchar's theory of mind, pain, and counting or calculation. And the idea is that it's the brain maps that correspond to Sacchar's and the brain maps that correspond to theory of mind are better separable in a way. So we thought, okay, fine, let's train a classifier and see if we can predict the task from the brain map activation data. So we used data from these 2 different databases. From brain map we had around 1,000 maps, and from NeuroSynth we had around 850 maps that correspond to these 4 tasks. We did not want to do any pre-processing, but the thing is, if you want to do classification, then you need to make sure that the data that you are training and the data that you are testing are on the same scale. So we did a very minimal min-max scaling, as in we made sure that the data in each of these maps is between 0 and 1. So, yes, as Julio said, there are around 900,000 voxels, and which is a dimensionality nightmare. So we reduce dimensionality in two different ways. The first is we removed all the 0 voxels. When I say 0 voxels, those voxels that have 0 activation among all 4 tasks. And then we did a PCA and took the top 100 principal components, and you can see the variance explained by these components in that maps there. And these 100 principal components explain most of the variance in the data. So we use these to fit the training data set. So see that little asterisk there? So time for some machine learning fundamentals. It's that whatever pre-processing or dimensionality reduction techniques that you apply on training data, you have to take the same and apply on testing data in order to make sure that the classifier does a proper job in identifying the labels on the test data. Yeah. So we used a linear support vector classification algorithm. Roughly what it does is, let's say you have 2 dimensions, x1 and x2, and blue and green, they represent 2 different classes. What support vector machine does is identify a separating boundary that maximally separates these 2 groups. Now, there is an issue with this. It is that if your classes are unbalanced, as in if there are unequal number of samples in your 2 classes, the support vector machines, they don't perform, or any linear classifier, they do not perform good. And the reason being, look at the black line there. If you do not specify to the algorithm that your class weight is not imbalanced, the classifier just tries to predict every sample as being a blue sample, which in turn boosts up your classification accuracy, but that's not what you want. So what you do is you balance these class weights to make sure that your new separating boundary or hyperplane is the red one, and it does a decent job in identifying both the classes. So that is what we did. We first took either the brain map dataset or the neurosynth dataset, and we did a cross-validation, a tenfold cross-validation to see if a support vector trained on brain map data, on 90% of brain map data, can predict the classes from the remaining 10% brain map data, and you loop this across all the data. And for the brain map, and remember there are 4 classes, so the chance accuracy is 25%. So we were able to classify brain map data with an accuracy of 60% and neurosynth data with an accuracy of 66%. So the next step is to expand it, as in we train on brain map data and we tested the classifier on the neurosynth data, and that gave us a 67% accuracy, and we did the other way around, and we got, those are the accuracy values that we get. So what that shows us is it opens a possibility for future studies to combine data from both these databases, train your classifier, and hopefully that allows you to decode fMRI data that you collect in your scanners. So we did not really get too much time to work on this, so I will only show the test data with 1 sample. So it's a binary thing. Yeah, for this we got a data set from Open Neuro database. Julio pre-processed it with fMRI prep, and it's from a study by Wu and colleagues.
That's
and
just pre-processed, I mean, not pro-processed.
I'm not even sure.
Although, well, maybe Julio became a pro in using fMRI prep.
That must be it.
So, the task is a thermal pain task, as in subjects had a pain block and a no pain block, and they had to, well, imagine that the stimulation is more painful than it actually is. And what we did is we took a bunch of samples from one of these pain blocks, took the mean, and subtracted the baseline, which is the first 2 scans in the fMRI run. And we trained the classifier on brain map data and tried to see if the classifier predicts that the test data, which is 1 sample, belongs to the pain paradigm, and it does. So again, it's a proof of concept, which we intend to scale later.
And then just a quick Outlook slide. So why does this matter? That you can train data using meta-analytic coordinates, basically, and then predict what your fMRI data was, which is what I just said. One thing that I think is a pro is that it doesn't require fMRI data to actually train. So that's something that just like jumped out to me in the machine learning talks, right? I don't have hundreds and hundreds of fMRI data, subjects worth of fMRI data that I can train classifiers on to then go predict something else. So here, this might be evidence that you can use meta-analytic data to train a classifier and then go straight into your data collection at the scanner. It could provide useful sanity checks during pilot scanning. So for example, you could ask yourself the question, does the classifier correctly predict my task activation data? So like if a grant reviewer comes back to you and says, how do you know that your task is actually activating a memory network or a pain network? You might be able to hack something like this to sort of get at that answer. One con is that classification could depend on training terms. So here we used pain versus saccades, which we think to be pretty different in terms of their spatial distribution. But if we train the classifier using things like pain versus touch or pain versus motor learning, we might not get such high accuracy. Those 2 maps might be harder to distinguish from each other. And the code, all the notebooks for it, and the data are available on GitHub if you guys want to download or contribute. That's everything.
And shout out to Katie, who is not here today, but well, I'm sure she's as excited as us about the project.
All right, on to MRI conception. Come over here.
Yeah, let's do it.
Can I just mirror?
All right, let's do that. All right, so you may or may not remember learning about MRI-QC earlier in the week, but this is a pipeline that takes BIDS compliant data and it runs some minimal pre-processing, and it spits out these really nice group reports that tell you about your different subjects or sessions in the data relative to each other. So this is a great way to identify outliers in your data, but when I bring this tool to people or show people, the main question is, okay, things like frame-wise displacement or full width half max, those have pretty easily interpretable metrics, so millimeters, for example, but what is something, how do we know whether these are good values or not? So this is where this project came from. So a lot of the IQMs, or image quality metrics that are calculated by MRIQC, have no sort of reference value. So there's no ground truth about what this number should be. And so this can be make it really hard to get a handle on what exactly you're looking at in the group reports. So our goal was to provide some context for the image quality metrics in the group reports by showing the distributions of image quality metrics for your data plotted next to a larger set of the anonymized image quality metrics pulled from the web API for MRIQC, where the image quality metrics are uploaded every time someone runs MRIQC, unless they tell it not to. So MRIQception takes the user IQMs for MRIQC and it plots them relative to image quality metrics from 200,000 plus images in the MRIQC web API. And so the user has the ability to filter their API query by relevant acquisition parameters like magnet strength, TR, TE. And another thing about MRIQC is that some of the definitions of the image quality metrics can be somewhat opaque for non-expert users. So to make it easier to use, we spent a lot of time trying to figure out how to rephrase the definitions of the image quality metrics in a more easily understandable way for lay users. And well, it's not shown as a tooltip right now, it's just shown under the plot. But we have that information too. So the goal for this is to basically make MRIQC easier for people to use so that doing quality control on your data, which is really important, is easier. All right.
Yeah, for some of the just back end of this, so with the scripting, basically we just had to have a way for the user to give this script their group's output, query the API and match it, and then compare. So first we're just kind of going through this. I don't think this is going to go. So you can like import everything. Do I have to push next? Cool. Wait, do I have to go down?
Nope.
Okay, cool. This is just defining our widgets, which we'll see later when we actually do the visualization. Cool. More widget stuff. Cool. More widgets. Did that work? I'm going to make sure it's new. Cool. Okay, so now what we'd like to do, like, so as a end goal, or have like the pieces here, is for you not to have to like pass a whole bunch of arguments. So we'd like to have like either buttons or boxes or a drop down or something. But what MRIQC has is either bold or T1 or T2. So for the demonstration, we're going to show you if someone wanted to compare their bold images from their group to the API. I do have to run it again. Should be cool. I haven't messed with that yet. And then here you can pick some of your parameters. So like we were saying, if you want to compare your group, you want to make sure whoever you're comparing it to or the scans match. Which one should I do here? We will do a TR max. What?
You can just go to the next cell.
Okay, cool. Nice. Let's go with this TR max of like 3. And then here.
While the mean works, and you can continue. While the mean works, the important thing is that you don't have to pick only one. So you could select a max of TR and a minimum of TR. And even then you can select a max of TR, minimum of TR, a max of TE, and a minimum of TE. So it works with several parameters at the same time.
Yes. I think this is just displaying it.
Yeah.
Cool. Okay, so this is where it starts coming. So we want to be able to like loop over one or many different of these variables that you want to visualize. This is kind of like part of the early hard coding, but those are all the ones that are available, and that's the way that they come out from MRIQC. This is, all right, cool. All right, so finally to the thing. So we're going to load in the data and just kind of show you like that it's working here. We have 18 test subjects here, and then they're on their test scans, and it has 45 different variables. And that's the user group that you've uploaded. And then here is where we pull in and filter the API information. And just to make this quick, we just pulled 1,000. So we're having some thoughts and issues, which we'll have Chris talk about querying the API. So for here, it's just 1,000 sample on 77 different variables. And you'll see that it then filtered down into 834 based on that TR filter that I did.
Yeah, so at the beginning of the week, I was kind of fighting the API, and I mostly lost until Chris Gorgolewski told me how to unlock the situation. So we still have to decide if we will query the API, because the syntax is MongoDB syntax, and it's a bit cryptic, and it would be difficult to ask the user to write MongoDB queries. So maybe so either we write a translation of your easy to understand commands to MongoDB, or we just download everything from the API. And the ideal would be to have a server that will update the database, and you could query this database and probably calculate the filters online.
So, and then just to group it, just to keep showing it, so there's 834, I don't remember how many we had originally on that. But then here, it then pulls these data frames, throws them together, gives it a grouping variable, so you know which was yours, which was API. That's why we now have 78 variables instead. And now to the actual, really cool part. All right, I'm going to do this, let someone else talk about it, but then we can visualize it. And
you have to zoom out again.
Yeah. Scroll, it's not scrolling. Control minus?
Yeah.
It doesn't like that. Command minus? Haha. Is that better? Cool.
Yes. So we made these plots using Plotly, which is a really nice package for interactive plots in Python and in R, and also in JavaScript if that's your thing. So we can select from the number of different metrics that MRI-QC spits out for bold images. So for example, frame-wise displacement. And then you can auto-scale. And so this shows the distribution of data points in your sample relative to the distribution of data points in the MRI-QC Web API data. And Plotly also has this nice feature where you can hover over various aspects of the image and it will give you some of the statistics. So this can give you a really good idea of whether your data are sort of normative in terms of image quality relative to other researchers out there in the world. So this is another example. This is the ghost to signal ratio, and you can see that down at the bottom it says ghost to signal ratio along the X or Y encoding axes. In this case it's at the X-axis, and higher values indicate lower image quality because there's more ghost than signal. So that was another feature that we added, was being really explicit about whether you're wanting to see higher or lower values, which was somewhat difficult to figure out for some of these metrics. And then this is just the temporal SNR. So. All right. So future directions for this is something we really want to, sorry, no. This is something we really want to continue working on and developing. Future directions are probably really improving the user interface, so in the way that Chris described, and also potentially having a better way for users to input their own data rather than going into the notebook and having to swap out the file paths. Other directions we've been thinking about, we had a lot of different ideas.
Like a cool thing would be like, let's say that you're scanning kids with ADHD, so your FD is crazy compared to it. If someone else could share their group of younger kids with ADHD, you could actually compare within your sample what might be a more realistic comparison rather than looking at 23-year-olds that are very, very still. So
yeah,
but that would involve people sharing.
Yeah, and you could certainly use, for your reference data, you could use the image quality metrics from the Abide data set or other data sets that are out there. Another feature we were thinking about was MRIQC has the widget where you can indicate whether the image quality of the individual scan is excellent, acceptable, poor, or just not okay at all. I forget exactly what it's called, but if we were to plot the individual data points on that gray half, it would be really interesting to have those colored by the expert ratings, which would give you another sense of, okay, maybe this is kind of a normative value, but that lots of people are seeing in their data, but those are also really bad images. So that might be changing our distribution. Any other things? Comments?
One final note, if you like really good memes, good work GitHub, because...
Yeah, I couldn't get them to load it. But they're funny.
All right, thanks.
Next is track funk.
So I didn't pitch this project, so that's why I'm numbered. I don't, it's just me. Okay, so I am tentatively calling my project TrackFunk. I'm open to branding advice and or recommendations should you have any. But basically the overarching goal here is to start incorporating information or data about function into analyses of white matter. This is something that typically isn't done because you don't usually have data about function associated with white matter. Okay. So for some time now, we've been building up a great deal of information about functional specialization in cortical areas, but there hasn't been any corresponding improvement or development of knowledge of what various white matter tracks might be associated with functioning. Every once in a while, I get a study that says, you know, maybe a particular property of a track is associated with a behavioral measure or something like that, but there's no real comprehensive set of knowledge regarding functional properties of white matter. So how can we deal with this? Well, as we're beginning to clarify various properties of white matter, one thing we can look at is the termination regions that are associated with those white matter tracks, so the various cortical areas that those white matter tracks are terminating in. So this is from a recent paper I did where I was looking at an understudied tract called the posterior arcuate and noting that it was terminating primarily in the posterior angular gyrus and then some posterior lateral temporal areas. So if we have that information about the cortical areas that the tract is terminating in, it seems that we might be able to leverage that somehow, given that we actually do know quite a bit about functional specialization of cortical regions. So what's the path forward? So what I did here at the hackathon was to just do a few of the steps that would be necessary to do this. So we already have some various segmentation methods for white matter. So you'll start with, as my advisor likes to note, a bowl of spaghetti. And it's a mess. We don't really know what's in there. We don't know which sorts of anatomical tracks are in that mess. But using, this is a method that we have available on BrainLife or, for example, AFQ, you can begin to identify streamlines that are exhibiting coherent anatomical structure. And so here, for example, you can segment out about 60 of these tracks. All right, so you've got your segmentation done. What you can do next is you can take each of those streamlines. Again, we're just trying to get model of what the actual anatomical tract looks like. And you can create a mask of the endpoints of that tract, because presumably those endpoints are in the cortex. So we're getting a sense of where that white matter tract is terminating in the gray matter. All right, so once you've done that, you then warp that subject space mask to MNI space, because that's where the information about the functions is sort of stored. And we can then leverage NeuroSyn to begin to go through the various coordinates associated with that mask and query NeuroSynth using the API to get specific loadings on coordinates and the various functions that are associated with them. So that's actually as far as I got here at the hackathon. What might come next is that some of those functions will probably need to be curated because they don't really make sense, given that they're like anatomical or something like that. It doesn't really correspond to a function we would describe. And then actually using that information to then develop some insight about those tracts. So for example, we saw the posterior arcuate there. That might connect regions that are associated with interaction with objects, given the various cortical regions that it's surveying. The GitHub repo is there. It's mostly a port of existing MATLAB code that I have on some other repos. So some potential extensions. One thing that's probably lacking in the NeuroSynth database is actual studies of white matter, given that it's mostly focused on gray matter. And so maybe trying to develop my own corpus of white matter studies and trying to distill some anatomical knowledge out of that might be worthwhile. Also, the code that is in that repo doesn't really work. It's just a collection of proof of concept functions, and I'll be trying to string those together into something usable and manageable in the coming months. But yeah, that's what I was able to do here.
Next is deep source.
Need a photo.
This one.
Is it? Do you have a HTML?
How about this one? Oh, you got that? No, the VGA doesn't work. So we want this. First, a photo.
Hi everyone. With Deep Source, it's got nothing to do with deep learning or DeepMind. This cool name. We wanted to run some simulations and compare different electrophysiological source reconstruction algorithms to see if, in theory, we could image subcortical regions in the brain. Aziz is not here, but he worked with us. So we chose to focus on the hippocampus. Just to give like a very broad overview, hippocampus is mainly studied in rodents, and there's not a lot of work done in humans at the moment. The work that is done is typically done in fMRI, because you can get to those deep brain structures. And it's a little fun fact, when you drink too much alcohol and have a blackout, it's actually your hippocampus kind of shutting down. So hopefully none of us will go through that tonight. Anyway, we wanted to see if, in theory, you could image this with another modality. And kind of, this is what we kind of measure in fMRI. We've got this hemodynamic response, so we might be able to see the hippocampus activity, but it's going to be on a really slow timescale. So we don't have any real kind of methods at the moment of seeing like kind of fast brain dynamics in there. MEG could be a solution to this. So instead of getting a sample every second or so, in MEG typically you get a sample 1,000 times a second. But it's a really sensitive technique, so much to the sense that if that's a magnetic field associated with a 7 Tesla MRI, MEG is way down here. So it's really, really kind of hard to actually do this in engineering. To give you kind of like a sense of the scales we're looking at, if the distance between me and you here is the size of the Earth's magnetic field, so is the size of an MEG signal, the Earth's magnetic field is kind of like the distance between the Earth and Venus. So really, really tiny. Anyway, what do we want to do? We're measuring some data here in sensor space, and then we want to map this onto brain activity in source space. Fortunately, we can do this with a good knowledge of Maxwell's equation, so knowing how the brain, how any electromagnetic activity propagates from the brain to sensors. This is actually Maxwell's statue with a seagull on top of it in Edinburgh. Just some terminology, because I'm going to mention this a few times. We actually formulate this with a lead field, which describes this current mapping from inside the brain to outside to this sensors, this matrix H. So we've got the sensor data, we've got the mapping, we need to go to source space. But this is actually pretty tricky, in fact impossible, technically speaking, for 3 reasons. One, your mapping is rectangular and also badly conditioned, so you're going to have mathematical issues. It's also ill-posed, which basically means there's no unique solution, there's an infinite number, and also you have 300 sensors or so, and you're trying to map onto about 10,000 different brain locations. So for these reasons, it's really tricky. And you can't forget the physics as well. Magnetic fields follow the inverse square law, which basically means that as you get further away from a source, it drops off of 1 over the distance squared. So you can see deep brain structures here, like the hippocampus, are really hard to image just because we're less sensitive to them. So with all these problems in mind, you have to introduce some kind of assumption, an optimization, and you can take 2 routes here. You can either take a tomographic route, which is a fancy way of saying that you're going to try and explain all of the variance in your data and minimize your model error, which is something which a minimum norm solution gives you, which we're going to talk about in a second. Or you can say, forget the variance, I'm just going to try and maximize my signal to noise ratio, which is what beamformers do. So, over to you.
So I'm going to translate what Ryan just said in Python code. But I cannot see. Okay, so for this we're using a package called MNE, which is like the standard electrophysiological analysis package in Python. So we are going to first create the forward solution, which is the H that Ryan described. Commonly, the forward solution needs a head model and a source model to be estimated, and for this we need MRI data. But luckily, MNE gives us an example of a forward solution, so we are just going to load that here. Then we need to simulate our source space raw data, and for that we need all these things that we load, but I'm not going to show you because it's boring. And we need to simulate our deep sources of interest. So in this case, we are saying we have an experiment. This experiment has different epochs, and some of these epochs are going to correlate with parahippocampal activity. This is what this code is doing. And this is our simulated data, a plot of our simulated data. So this is our grand truth. Like the activity during these epochs should be located here, right? So from this, we're going to simulate sensor space data.
And when we simulate sensor space data, what we do is basically create sinusoidal waves based on that source space. And then we also add artifacts, so we add noise, we add EOG artifacts, so eye movements, and we add cardiac artifacts, which are the most, so the artifacts that contaminate electrophysiological signal the most. And this is how our raw data looks, right? So now we can go and see how the source reconstruction methods work. And so first we compute the inverse with E-Loretta, which is kind of a minimum norm estimate. This is the code, and what you're going to see, it's really depressing. This is hippocampal activity for computed with this source reconstruction method, so it's all over the brain. Context, my PhD is based on seeing hippocampal activity with MEG, so it gets better, yeah. This is computing the inverse with the minimum norm estimate. This is the code, still super shaky. And then this is with beamformer, which is like the right side of the path that Ryan just explained. This is the code, and it seems to work a little bit better. Not perfect, but a little bit better. But then there are some tricks that we can do to the data, and we can do some mathematical, more complicated stuff that Ryan is going to explain that he did in MATLAB. I'm sorry. My job is going to be to translate that later into Python. Yeah, sorry. Oops, how do I exit this?
Okay, so Martina just showed kind of one example or one instance of a source reconstruction algorithm where we wanted to kind of test over many instances and different signal to noise ratios how good a source recon algorithm works. In the first case, what we did is we simulated a source in the left motor cortex, so kind of high up, so it should be quite visible for MEG data. And then we made 3 minutes of data 5,000 times and tried to source reconstruct activity onto this part of the brain on an 8mm isotropic grid. To make this a bit harder and slightly more realistic, we added biological noise, shown in the black dots here, which varied over iterations, and also added sensor level noise, like Martina just said. And shown in green here is the ground truth location, and then in the red and yellow is the power reconstructed. This has been thresholded, so a bit of massaging, but what we then did, we wanted to see how accurate this localization was, and so we said, what's the most correlated source with what's the distance between the reconstructed source and the ground truth, the maximum? And what you can see is that for superficial sources at least, the gradient is much stronger this way, which basically means that you're more sensitive to biological noise rather than sensor level noise. So you need your source to be strong in source space. That's just one metric, and just saying, okay, we found the maximum isn't perhaps the most holistic way of saying my algorithm worked. So we also looked at the full width half maximum of the profile in the source space as well. And interestingly, actually the kind of opposite is true. So the gradient is stronger this way, which means that your spread of your reconstruction is more sensitive to your sensor level noise. You have to take both things into account. Anyway, we're interested in the hippocampus, so the next step was to go deeper into the brain and put a source in the hippocampus and see what we got in that case. And here, you can do it, but it's harder, and you get more of a spread of power in your solution space. And then these numbers are actually slightly larger, so your reconstruction isn't as good because you're not as sensitive to the deeper sources, but you can do it. And we've kind of shown this in simulations. Just a note on like the kind of open science. Our code is available on the GitHub. Most of these simulations were written in MATLAB, and that's kind of par for the course in the EEG and MEG community. It is catching up, moving to open source stuff like Python, but also the open data sets, the fMRI community are really kind of leading the field. This is mainly because MEG data tend to be a bit more heterogeneous, and you have to do a lot more kind of harmonization. So yeah.
Yeah, so we end up showing you our GitHub repository that we have created and the things that we want to do next. The first thing I would like to do is fix my binder. I broke my binder launch. Yeah, something to do with the environment YAM. And then these are the next steps that we are going to do. We're going to tidy up our Jupyter notebooks because they look super messy right now. Then we're going to incorporate and compare other source reconstruction methods. We're going to add a very fancy source reconstruction method based on hidden Markov models. And then all this was qualitative comparisons except what Ryan just showed. So we are going to quantitative compare all these things in Python. And we are going to simulate ground truth data varying all these things. So yeah, if you want to contribute or if you're interested, you can just come to our GitHub and see what we are doing. Thank you.
Next is
What else?
Would you mind being live tweeted? That's fine. Come stand in front of us.
Alright, so we're PyCortex Edu. I'm Armand, this is Tal, this is Edward, and Matan, and we also had a bunch of help from Alex and also Tal for PyCortex and NeuroSense respectively. So the motivation for our project is that there remains a need for an all-encompassing brain educational resource and one solution to that is basically an amendment of already existing 3D visualization software. And so what we did was we went about trying to get PCA variance components, which explains most of the neuroimaging literature, as well as some of the NeuroSynth maps into this 3D visualization tool, as well as parcellation schemes to kind of see how that relates and also just have it all encompassing. And so the process to do that is we basically linked PyCortex with NeuroSynth, and then we applied PCA to NeuroSynth results, as well as kind of kept the maps to visualize by themselves. And then we also created label files associated with an alternate atlas or parcellation scheme, which we want to make more of. And all of this was interfaced together with amendments using JavaScript to PyCortex. All right, so this visualizer is live. You can go to this URL right now on your computer to see it.
Look at this URL right now.
Or maybe not all.
Although, small note, if you're in Chrome and you go to the URL, there's a shield icon at the end of the URL. You have to click that and then allow unsafe scripts. But I promise it's safe.
That's my fault. I didn't want you to fix it.
Okay, so as you can see, we have this pretty and a colorful brain. This is FS average, and the colors actually do mean something. So if you go to the top right and go to the controls, you'll see all these PCA components. Before I click through it, I just want to show the interface of PyCortex. So you can flatten the brain. Whoops. And you can inflate it. And we have an atlas. So if you press L, it displays all of the gray matter labels. So what's showing right now is the top 3 components. So if we take all the keywords from NeuroSynth and get a map for every keyword and then do PCA. The top PCA component was related to motor. So at the bottom right, you can see the top 5 terms that were associated with the component in the bottom 5. The second component was language and semantic. And the third was auditory. Fourth was visual. Fifth was amygdala, hippocampus. 6 was default mode network. If we take just the top 3 and we give them different colors, that's what produces this colorful map. And Edward will show what happens when you click the brain and the left side.
Yeah, so for example, if you're interested in studies that are associated with this, then here is you can click on associated studies. See if that works. No, maybe. Or you're interested in what the regions in the brain are associated with alcohol, for example. Then this pretty map lights up, and then you can, for example, also make it a little bit more clear. And like, oh, what happens in this brain region? And then, well, what it should do, it should load something from NeuroSynth, and you can click to the studies associated with that brain region. And you get also the terms from NeuroSynth. You see what's linked to that brain region. And we also filtered out all anatomical terms so that you don't get confused, because otherwise you see strange regions that are from NeuroSynth associated with that area you're at. So that's about it, I think. Thank you very much. Did we use like the shields?
Oh, the shield.
Yeah.
It's the shield.
It's a shield.
Yes, it's very addictive. That's right.
Yeah.
Next is the psychiatric phenotyping.
And in the meantime, we'll show that it works.
Can we see it before we go? Yes.
That was
there
working.
we go. So, keywords on the left, you can click it, it loads the map. Bottom left is studies.
Okay.
So our motivation with this project was to...
Sorry.
I'm okay with it. Yeah. So our motivation with this project in this week was to come up with a way to decide which behavioral measures are most promising to study any given psychiatric disorder. Because if you just ask psychologists or psychiatrists to come up with which measures to use, you cannot get people to agree because they come from different theories and they have different views on how the mind works. But then we wanted to take a data-driven approach to see which measures better differentiate people with a disorder and those who don't. And in the beginning, we ran into a problem because the only data set that we could access, in this week at least, didn't have many people with a psychiatric disorder. Actually, we focused, like as the title says, we focused on the Human Connectome project data. And even though they specifically wanted people more or less healthy, apparently 25% of the people there, they had some sort of substance abuse disorder, which is quite high, but it allowed us to go on with this project. So we focused on substance use disorder, and there are many measures that one could take, but we cannot, in practice, right, if we are interested in developing something for the clinic, we cannot have a person taking more than typically 10, 12, or something like that measures. More than that, you need a lot of time, and this is simply not feasible in everyday clinic. So we wanted to minimize patient burden, but without sacrificing some sort of predictive accuracy. So I used the Human Connectome project data that had about 1,200 people, and we focused only in behavioral measures, and specifically measures that could be taken not only in the office, but also potentially online, so that you could automate those measures and make the whole process easier. So we had 293 people with a diagnosis of substance use, or at least that, according to the DSM criteria, had a substance use disorder. So this is the correlation matrix of all the behavioral measures that we got, about 73. And there is a big cluster here, but this is essentially, it's the same task, but different parameters that you can get from the same task. Is a task of impulsivity. Other than this task here, there are some correlations that appear in the data, but it's not extremely correlated. So they are really measuring different dimensions, but maybe you don't really care about all those dimensions, right? So you wanna jump in?
So, the behavioral data alone has 70-something measures, which if you're in a time crunch and need to determine if someone has a substance use disorder, then you're not going to want to have them complete all 70-something. So we used models to sort of narrow down which of those measures you would want to use, and we did that via feature selection. And for those that aren't familiar with feature selection is, just imagine you've got an array of measures and you're just trying to figure out which ones are most predictive of your outcome variable, which in this case is just ones or zeros for being classified with substance use. So what you do is you sort of select, these models select the few most important ones, and you can sort of use those to get a sense of what kind of accuracy you can achieve with the subset of data and the labels you have. So for this, we use scikit-learn's recursive feature elimination algorithm, and we pass through logistic regression and support vector machines. I want to thank MetaClass for explaining what linear support vectors were, so I didn't have to. So those were the two models we chose just because we have two classes and we're trying to keep it relatively simple. We also tried lasso regression because we learned about that in the scikit-learn tutorial, so why not give it a go? But this not really being our field, we don't really know what recursive feature elimination is doing. It's supposed to be dropping a feature each time until you get to one left. But we found this great graphic that sort of gives us a hint at what feature selection is doing. So it's essentially you have all these features starting out and they sort of like drop off the further and further you go until there's like one left. This is apparently done in some sort of genetics lab or something is where we grabbed this graphic from. But we think it's pretty representative of what we think is going on in this black box. And the more you watch it, the funnier it gets. Because you just like want the others to do well. So we ran this recursive feature elimination algorithm. Essentially, we ran 2 logistic regressions and 2 linear SVMs, and the reason for that is we realized there's 25% substance abuse diagnosed people in the set and 75% control, so we were worried about the imbalance in N because when we tried to run just like classifications with like 1 or 2 features, they would always just classify everything as a control and get 75% accuracy. So we ran models with all the data as well as an equal N for the 4 groups. And what this shows is, so all the way on the right would be if you're using all the features to try and predict the labels, and then moving to the left, you drop features as you go, one at a time to try and get a better, to try and obtain a better accuracy score. And the optimal features pulled out from the 4 models vary slightly, but generally you can get less than 15 out of the 75 to have some like 60-something% accuracy, which isn't great, but the people in the field have said it's not bad, so we'll take it. But there is a little bit of variability in the models themselves based off of like how you standardize the data and stuff like that. Here's our lasso regression attempt. This is all thanks to Teddy. We found that you can sort of see, depending on the alpha you choose, you can sort of select how many of the variables you keep toward the end, and that sort of gives you a way to sort of stop when you reach a certain number of features remaining. You want to sum up the results? Someone go.
So from that, because we don't really have a strong reason from selecting one model over the other, then we asked which measures overlap between those models. And then, as you can see here in this frequency diagram, a few measures are always present, and we do have some significant overlap, although there are also some measures that are model specific. And with that, we came up with this word cloud of which are the most informative tests, and the size shows the tasks that are that appear more often across models. So as you can see, delay discounting seems to really be consistent. So if you want to work with drug abuse, then you probably should be using this task. But also we have some interesting patterns showing up that we have lots of personality measures and a few measures of executive function / fluid intelligence. It has given us a framework, not only, I think, just one of us actually works with substance abuse, but it has given us a framework to how to tackle this problem for different disorders. And that's it. Thank you.
And last but not least, the TLS-DTI bridge.
I wonder if someone can connect to it, or if someone will like this.
Do you need to connect?
Oh.
You have an HDMI? What do you have? I have this kind. Maybe this is USB-C. Does somebody have a converter? It's sort of like a previous generation, looks like a previous generation Mac to HDMI. Just, do you have just PDF? Or?
Yeah, it's just PDF.
Maybe Slack me the PDF and we'll connect.
It's on the Google Drive already.
Okay, great.
Because I got like a bunch of ones from Max.
I think we're actually.
Oh, okay. Okay. So I know that everyone. Are you gonna tweet it? Yeah, you can. I know that everyone is very tired now and looking for a boat, but so I'll try to make it short. And my project was about bridging diffusion data with TMS data. And that's probably the only one slide where you will see my results visualized. Yeah, these images are from actual data. So why I wanted to do that? We were speaking a lot about replicating studies and so on. And the idea about this is kind of stems from this paper where these researchers were trying to model TMS effects for realistic fiber trajectories based on diffusion imaging. And the idea behind is that the effect depends on the actual geometry of the fiber you stimulate, and there are at least several mechanisms that are involved. Well, here I put 4 mechanisms, but actually there are 3 major mechanisms. That there are a particular mechanism that is related to sharp bends of the fibers, the one where you have lambda squared over the directional derivative of the field is just related to the cable equation of your axon. You have also transitions between gray and white matter where you have changes in conductivity. That's another source of activation of fibers in TMS. Well, and at the terminations of the fibers as well. And if you just try to get your field within your brain and calculate those effects on some fibers, you will see that the fibers, even if they're located in a similar, very close to each other, they may not be activated in the same way, because some of them are bending more than others, and while others are just going straight. And so my idea was to get something like what you see in the bottom right corner. And that's one point of the story. The other point it's familiar for those of you who are working with diffusion. You know that different strategies could be used to generate tractography results, and depending on the strategy you use, you may get very different results. And this is the same subject of my data set, exactly the same region of interest, but 4 different strategies. And you see, well, here it's supposed to be corticospinal tract and corpus callosum, and can you see that the results look very, very differently. So under A, it's the usual DTI tensor model. And you're not able to get any lateral branches from the corticospinal tract and from the corpus callosum going to hand regions, for example. That's what you can typically see in papers based on diffusion tensor model. On the right is cubal model, under B, on C is spherical deconvolution, and on D, the results that I had after running those data in Traxec. So all of them look different. Well, and even within the same model, if you change your parameters of the model, the turning angle, the step, the results would look different. And there's no way to see which one model is the best. You may try to validate it using phantoms, but you may try to validate it using functional imaging, functional TMS approach. So that's the general thing that I was doing during this week. So there is a software called SimNibs that allows you to simulate EF fields within your brain based on individual anatomy. And so far I was taking the result of that. You see those mesh with the colors showing the amplitude of the field. And on the bottom, I was taking diffusion data, and Daniel showed me how we can use brain life to generate many different tractography data sets, changing their parameters. And then what we made, we just combined those output of the SimNibs and tractography data to calculate the results. So and so far, I've out of these 4 mechanisms, I've implemented 2. The one that takes into account the geometry, the bends of the fibers, and the one that is related to the cable equation with lambda squared. Because so far I didn't take into account segmentation, white matter segmentation, I didn't look for the third mechanism, but that could be a next step. And please believe me, I've generated all those results for each line, but surprisingly this morning I found it very tricky to make 3D visualizations. So to show a kind of exact effect at each point of a fiber. So that would probably the next point. I thank Christy very much because she sent me a magic command line, which I hope will help me to do that. And the next step would be to use actual TMS experiments later, and there you can calculate, measure and quantify those effects, actual effects in your experiments using Heatmap software, and then to see which of the diffusion models would be better explaining the actual results. That's one point. And yeah, there's many things that should be improved here, and if you would like to participate in this project, there is the link to my project. There are 2 things there, the script that actually makes those calculations, but the problem is that it will not work on your computer because it requires some very weird environment. And that was the problem on Tuesday when I was trying to run it on my local machine while my remote computer has been shut down. And all Tuesday I was trying to build a Docker container, and I didn't arrive to a point to build it. I created many Docker images which resulted in the fact that I've lost all my space on my disk. and all night I was cleaning my disk from all those Docker images. Yeah, if someone would help to do that, that would be the first step for further collaboration on this project. Well, and then we can look for more funny things, just try and say, finding an optimal point for stimulation so that it would produce the biggest effect. So those people who are fans of optimizations, you're welcome here. Oh, that's it.
Okay, great. I think those were all the different projects. That was amazing. So much you can do in just a few days. And all that remains is to celebrate. So our boat actually leaves